we have mental disorders which may share specific genes, which I mean is like the same gene has an effect on both or multiple genetic disorders like schizophrenia and depression in our case. So what we did is what is called the GWAS, which is a genome-wide association study, which estimates a p-value and effect size, etc., for each position in the human genome, and regresses them essentially on depression or schizophrenia. So we get an associated p-value for each position. And then we basically split the genome into blocks. And I will come to this later. And then we have some data sources, which are ones we took from the PCG, which is like Saturated Genomic Consortium Studies, which is like a basically summary where we share our genetic data. And the LD blocks come from this website, which I come later on. So yeah, this is how the data looks like if we download it. So you get like, these are the two studies. We only looked at depression and schizophrenia. And so you got like what we call RSID. So these are identifiers in the human genome, which are basically IDs in the position of the human genome. And so these are basically, we can merge with those. And then we have like alleles, like alternative and reference allele, which is, if you remember high school biology, we have ACTG, which are the 4 letters of the human genome. And so we have, like, let's say, for example, the first row here, we have 1A, which is C, which is the less common allele, and we have G, which is the more common allele in the population. And so you would assume, for example, if more people who have schizophrenia would have the C allele than the G allele, that the C allele might have a negative effect on schizophrenia. And then we have like some coordination in the human genome, which are like, these are coordinates in the human genome, and a bunch of other information which are not necessary. But the most important thing, we have the p-value associated with schizophrenia and depression. Yeah, that's like overall data. So now we come to the blocks. So human genome is just, they're not unrelated. Each position is actually correlated with each other, and therefore these what we call LD blocks. And as you can see here, for example, this is an example. Here we have 2 LD blocks. And we can form these LD blocks with some techniques, which I won't go into detail, it's more complicated, but essentially these are blocks which we hope are independent from each other. And so our approach was essentially, we look at each block and look at the p-values in these blocks, and look how that p-value inflates from the expected distribution of p-values if there would be no effect. And so we did this for each block in chromosome 1 because we run out of computation power. So this is how it looks, however, for all the positions in the human genome. So the blue one is schizophrenia and the red one is depression. And as you can see already, schizophrenia is inflated, which means there's a very strong signal in schizophrenia. In compared depression has only a tiny signal here in the end. That has multiple different reasons. One of the reasons is schizophrenia is more heritable. Like, we know there are bigger genetic factors in schizophrenia than depression. Heritability of schizophrenia is around 80%, depression is around 50–60%. So you would expect already like there are more genetic factors in schizophrenia. And also schizophrenia might be more polygenic, which means like more positions in the human genome are important for schizophrenia than depression. Or the other reason is we don't have enough power. Maybe the sample size in schizophrenia is higher than in depression because it's maybe easier to get good phenotyping or good quality data in schizophrenia than depression. And one other reason is that, for example, we have very clear definition what schizophrenia is, but major depression is a bit more fuzzy. That can be another reason. So the variance or the overall distribution, for example, of depression might be more I don't know, less, has greater variance, for example, than, for example, schizophrenia. That's like very different reason there. So yeah, and then basically we looked at each plot and basically computed what's called an inflation factor. Basically we looked, for each plot we computed these basically kind of graphs, and then estimated how much. So the line here is the expected, if there would be no effect, and we basically estimated the inflation factor of that. I won't go into detail what that inflation factor is, but essentially it measures the strength of the association in this area. And then we compared each block with each other. So here on the x-axis we have schizophrenia, and on the y-axis we have major depressive disorder. And these are like, so inflation factor of 8 means this is a very strong signal. For example, here in schizophrenia we have here a very strong signal in one of these blocks. So like inflation factor nearly 8, but like in depression it's basically nothing. There's no inflation. But for example, here we have an inflation factor of 2.7, I think, but it's hardly anything in schizophrenia. So these are disease-specific signals. But this one seems to be shared. This one block seems to be shared across both disorders.
You've got a lot of blocks. How do you know that that one block is actually special and not just an artifact?
You mean like in total for like, well, you mean like just random?
Just random. How do you know it's significant?
Because we compared it to the expected distribution initially. So these inflation already estimate what would we expect if there would be no signal in that block. Yes, it can be random. Overall, maybe some of these inflation blocks could be random. We haven't tested it yet. We could do that later on. Yeah, maybe. Okay. But I mean, an inflation factor of 8 is really high. It's unlikely that it's completely random.
Okay, and it's high even though you've got a lot of blocks. I
Yeah.
mean, normally the significance would depend on the number of blocks that
Yeah,
you've got.
yeah. I mean, you could probably like take the distribution of all these signals and see what the distribution is, and that would probably vary end of the tail. Yeah. I mean, we have actually plotted these plots, and yeah, these are the 8 blocks that really fat-tailed far out. So I mean, to come to the conclusion, there seems to be some genomic region which might share effects. It's a very quick and dirty way, to be honest. I wouldn't publish it in any form, but there are a lot of stuff you can do. There are a lot of caveats as well attached to this. We had little computation power. We only investigated chromosome 1. Sample of both studies might be overlapping. The controls, maybe the controls were used in both studies, so that might bias the results. We might have missed a number of things by only looking at blocks and not doing something more sophisticated. And maybe inflation factors are also not a good measurement as well, and there aren't. So there are a lot of things, but it's a short afternoon, and we just took them.
That's good.
Any more questions? Okay. You want to say something?
Yeah.
Oh no, for the other project.
Yeah.
Yeah.
No. We should now.
Okay. Just can't say that we did much today. Probably it was just a good experience.